ice because it needs to be cold the entire time. You can't let it sit at room temperature. Okay? Then you'll have these 1 ml pipettes, and they are sterile. And when you open them up on the end that has the cotton in them. Okay? And then you're going to use a pipetter. And then for 250 microliters, this is 1 ml if you go all the way to the top. Okay? So 250, and don't touch it like I'm going to do right now because it won't be sterile, but I just want to show you. The first thick line that has a number is 1 or 0.1 or 100 microliters. The second line with a number is 200 microliters, and between the second and third number is going to be 250. Okay, so you're going to pipette up to 2.5. Okay? So this has been sitting on ice, but I don't have any liquid in there, so I'll use one that I have liquid in. So pretend that was sitting on ice, so it's ice cold. And I want to pipette 250. So I'm going to go up between the second and third number. And then I'm going to put that into my new control and ligation tubes. And you roll down. Take another 250. If you notice when I'm pulling it up, I roll it down where the plunger goes up. When I dispense it, I roll it down where the plunger goes down. Okay, or up where the plunger goes down. I don't know if any of that just made any sense. Okay, so I have I'm on step 3. I have my 250 microliters of each solution into my new ligation and control tubes. Now it says pick a colony from the source plate. So now we have the plate of bacteria that has source on it, and we're going to pick a colony. And these little guys that look like hockey sticks are for picking colonies. Okay, so they look like toothpicks, plastic toothpicks that look like hockey sticks. Okay, do you see that? You're going to pick your colony with the pointed end, but don't gouge the agar. Okay, if you gouge the agar, it will mess up the experiment and you'll end up with just agar in your experiment. So you want to gently scrape off a colony, and it says pick two colonies 2 to 4 centimeters or millimeters in diameter. So pick a couple of colonies, don't overdo it, but don't gouge the agar. Okay, then you're going to take those colonies that you picked, put them in the tube, and it says to turn it. So you're going to, with your fingers, just turn it back and forth, and you can see and that's just to get them off of the hockey stick so that they can go into the liquid. Okay? You would have some sort of a waste container for the bacteria. That's where you would put this in instead of on the table. You would get a new hockey stick and pick a couple more colonies. Once again, do not gouge the agar. Just scrape off the top. Put it in the other tube. I put the first one in the control. I'm putting this one in the ligation. Okay. Once again, you would dispense of it into a waste beaker, but you wouldn't set it down. But I'm not using bacteria today, so I'm setting that down, but you would not do that, okay? Now, it says suspend the tubes by tapping or vortexing. So once again, turn this guy on like that, and you don't want to do it too long, just enough to get it mixed, okay? Now you're going to take the reaction product from module 1. So remember, we put the solution in these 2 tubes, let them sit for an hour. We have one labeled control, one labeled ligation. So you're going to be taking the stuff from the control from the previous module and putting them into the tube that you just put the bacteria in for the control. And then take the ligation tube from the previous module and put it in the ligation tube for this one. Okay, just try not to mix them up. So now you're going to add 5 microliters. So to do 5, we're going to take this down to 0.50. Okay, remember, always go down and not up. You don't want to go over 2, but you don't want to go through 0 either. Okay, so we're at 0.50, so that's 5 microliters. Not very much. Okay, so we're going to take 5 microliters of the stuff from module 1 and put it in the stuff that we just made in module 2. Okay, we're going to dispense this tip and get a new one, but I'm not going to waste. And you would close those. 5 microliters, is not very much. Okay, very, very little. And you're going to put it in here. Okay, once again, we're working with bacteria. We're going to dispense our tips into that waste beaker. Okay, once you start working with the bacteria, you have to dispense everything into that waste beaker that's going to be bleached. Okay, now it says incubate tubes on ice for 15 minutes. So now we can take the tubes that we just put them into, and we put them in our little ice water bath, and you push them down to where they actually go through the foam thing to where they're down in the water, but they're going to be down in the ice water. Okay, there we have a 15-minute incubation period. During this 15 minutes, you can prepare for the next one. Make sure you read and know what you're going to do. Label anything that needs to be labeled. Okay, so we have a 15-minute period. After that 15-minute period, step 8 says place the tubes in a water bath for exactly 90 seconds. So we're going to have 2 water baths set up. And they're going to be on the table or on the side where we usually have the incubators, I think, unless they're on the other side. Okay? One is going to be labeled 42 degrees, and one is going to be labeled 37 degrees. Okay, 42 degrees is pretty hot. 37 degrees is body temperature. So you're going to take your entire thing of ice, walk over to the water bath, take your foamy guy out, set it in the water bath, count 90 seconds. After 90 seconds, you pull it out of the water bath and put it directly back into the ice, from the water bath to the ice. Okay, then it's going to sit for 2 minutes. Now I'm on step 10. It says using a sterile pipette, use 250 microliters of recovery broth. Okay, this is the same pipette, but you would be using another one. Okay, once again the same amount, 250 microliters. Now the recovery broth will be in a bottle about this big, or it may be aliquoted for each group. I don't know, okay? But anyway, let's just pretend this is recovery broth right here, and it's going to be broth color, like that amber color, okay? This is a new pipette. I'm going to get 250 microliters. And I'm going to put it into each of my new tubes that I just had on ice. Now I'm going to take them out of ice now, because it was on ice for 2 minutes. I take it out of the ice. I add the recovery broth because it's at room temperature. Okay, and I take another 250 microliters. Put it there. And now those guys are going to sit at room temperature for 30 minutes. Okay, now we have another long wait period. So during the 30 minutes, whatever you didn't finish in the 1-hour incubation, finish now. Maybe you're not done with your oxygen lab or something like that. So do that during this 30-minute wait. Okay? After that 30-minute period, it says place the tubes in a microcentrifuge. I'm sorry. Stop. Okay. Okay. So you put them in it in the recovery broth. Okay. And then you incubate it at 30 degrees, and I missed a step. Okay. Oh, man, I'm misspeaking everything. Okay. When you incubate them for the 30 minutes, you're incubating them in a 37-degree water bath. So if you read step 11, after you add the recovery broth, you don't let them sit at room temperature for 30 minutes. You take them and put them in the 37-degree water bath for 30 minutes. Okay? Then you're going to be doing whatever else you need to do. After the 30 minutes, you take them out of the 37-degree water bath, okay? And then you place them in the microcentrifuge. Remember, balance them out. Several groups would use one centrifuge. Now you're going to turn it and you'll have to time for 5 minutes, okay? So you want them to spin for 5 minutes. After they've spun for 5 minutes, then you're going to take them out, and what you'll have is a pellet of cells at the bottom and then all this liquid. And what you want to do is take out 0.4 mils of the liquid. So using a new sterile pipette, 1 mil pipette, you're going to take 0.4 mils, which would be 1, 2, 3, 4 numbers up, okay? And you want to make sure you have a pellet. If after 5 minutes you do not have a pellet, you need to put it back in there and spin it longer. Okay, you have to have the pellet or it's not going to work. So now I have my pellet at the bottom, and I want to take 0.4 mils of the liquid, which is also called a supernatant. Okay, and I only want to take 0.4, and I don't want to take any of my pellets. So make sure, even if you don't get 0.4, make sure you leave your pellet in there, okay? And then it says to discard this, that same waste beaker that we put our little toothpick in and our E. coli tips in, we'll put in there, but I'll pretend this is it because I don't have that waste beaker here. You would do the same thing with your control one, take off the 0.4. Now, it says to resuspend the pellet. So whatever liquid is remaining in there, you take that same pipette and go up and down, up and down, and up and down. Okay? Now, I want you to notice on the next page, it says obtain the 2 auger plates. So while you were in that 30-minute wait period, you would have gotten 2 plain auger plates, and you would have labeled them ligation and control, okay? And then with your group name. Now you're going to take 0.1 mil of the stuff you just made where you took the supernatant off, then you resuspended it. You'll take 0.1 of that, which would go up to that first number, and you're going to put it on the plate, okay? So the best way to do this would be to, when you take your tube out, remove the 0.4, discard it, whatever's remaining, go up and down to mix it. Using the same pipette, pull out all of it. No, 0.1 mil, okay? Put it on your plate. And put the lid on, okay? Now get rid of that tip, that pipette. Get a new one for the other one, okay? Do the same procedure. That way you're not going back and forth trying to get different pipettes, okay? And these are all going to be put in the waste beaker because they have bacteria on them, okay? Then it says using a sterile loop. So then you take one of these sterile loops. They're provided with the kit, so we're actually not using our flames, which we could, but these are provided, so we'll use them. So these little yellow loops, which I cannot get open. Okay, it's going to remain sterile forever. All right. So using the sterile loop, you don't want to touch the loop side. The 0.1 mil, you're just going to spread it, kind of turn it and spread it, and just spread it all over the plate. Okay, you'd use a separate loop, a different loop, to spread the other plate. Okay, you can't see this because all this stuff is on me. Okay, so I'm taking and I'm spreading, I'm turning. You're just spreading it all over the plate. You just want to get as much surface area as possible. Once again, this is contaminated with E. coli, so I'm going to put it in that same waste beaker that I've been putting all the E. coli waste in. Okay? For each plate, you would use a different loop. For each tube, you would use a different pipette. Okay? And then it says, allow them to absorb for 15 to 20 minutes. So what you do is just put these off to the side. Depending on what time it is, you may have time to wait the 20 minutes and then put them in the incubator. If we start running out of time, I'll stay the extra and put them in the incubator. Okay? So in the meantime, through all of this, you need to have finished the oxygen and the growth of bacteria lab. As you can see, this procedure is very long, very time-consuming, and very precise. Even I was making all kinds of mistakes as I was going through this. So it's very, very important that you study about the procedure before we do it and be absolutely prepared to do it and read the procedure. Okay?